So we will move towards fungal classification. As you all know that fungi, uh, there are different behaviors of fungi as far as their nutritional types are concerned. So one of the type is the symbiotic fungi. So basically what uh, they contribute in your daily life actually, and they make relationships, mutual relationships with the plants, which are the primary producers, and they help the plants in absorbing the nutrients from soil and absorbing the minerals from soils, and in turn, those plants which form relationships, mycorrhizal relationships with the fungi, and they grow better than those which lack such uh, symbiotic relationships, because their capability to absorb water and minerals, it is greatly enhanced after forming mutual relationship with the fungi. They are the primary decomposers, right? Uh, along with bacteria. Okay, but if I compare the two groups, fungi, they are much more uh, potent decomposers comparative to the bacterial counterparts, or comparative to the bacteria. So you cannot imagine this world without these uh, decomposers. Okay, otherwise there will not be any place left for the human or for any other creature on this planet if the decomposers they are not present in the environment, right? So this is how important they are, right? uh, Plus there are different ants which cultivate fungi, and these ants basically they cultivate fungi, a form of relationship, symbiotic or mutual relationship with the fungal strains. So the reason for this is that these fungal strains, they are very much capable of degrading cellulose and lignin from plants. So after that, glucose is formed, which is utilized by these ants. So there are a lot of ways through which these fungal strains of fungi, they are contributing in your environment. Plus, as a food, these fungal strains are used. Mushrooms, they are one of the fungal groups that are used as a food. Plus, you have heard about Saccharomyces or baking yeast, which is used in the baking industry. Plus, fungi is a source of a lot of antibiotics, and it's also a source of alcohol. So many industries, they are basically established, and they are running smoothly because of the activities of these uh, fungal species. Right, but generally, apart from these beneficial effects of the fungi or fungal strains, mostly uh, fungi is assumed to have a negative impact on our mind. But if we have a look at their statistics, so there are a lot of species, like I have written here, that more than 1 lakh species of fungi they are reported, but only 200 of them they are pathogenic to humans and animals, right? They may also spoil the different things, your food, your infrastructure, but again that a lot of positive contribution is also uh, being continuously done by these fungi, right? So if you are looking the properties of the fungal strains or fungi, basically what is fungi and what is its structure, right, and how it look like. So basically, you all know that a fungal body is known as mycelium, and mycelium is composed of very fine thread-like structure, hypha, and they are chemoheterotrophs. And chemoheterotrophs means definitely they are not able to synthesize their nutritional requirements from sunlight or from any other source of energy. Rather, they decompose the things. They are the chemoheterotrophs. So they make the things simpler. They actually degrade the complex things into the simpler ones, but they cannot synthesize the raw material their for completing their nutritional requirements. So they are the chemoheterotrophs, and they are aerobic or facultatively anaerobic. So they require oxygen, and if mostly they are aerobic. Few of them they are facultative anaerobes, which can survive in the presence of oxygen. And only a few anaerobic fungi are known. So we'll discuss on the WhatsApp group that what are those types. And there are fungi can be seen into two life forms. One is the mold form and one is the yeast form. So yeast basically is the unicellular form of the fungi, and mold is the multicellular form of the fungi. But one thing is common, whether it is yeast or it is mold, both are eukaryotes, right? So if you're talking about their identification, that how you identify that which yeast is there and what is the species of fungi you are dealing with. So you need to have some biochemical test results, right? So particularly yeast identification can be done by using different biochemical tests, different sugar tests, other biochemical tests, but the molds, they are identified on the basis of their colonial characteristics and the spores they produce. So as you can see that there are two types of hypha. Either it is the hypha which is divided into the fractions or the fragments or compartments by means of a ball-like structure which is known as septum. In that case, it is called as septate hypha. And if it is not present, then the entire hypha is without any compartmentalization. So in that case, a lot of there are a number of nuclei in the hypha, and such hypha is known as cenocytic hypha that contains multinucleated structure, right? So multinucleated hypha is also known as the cenocytic hypha. So the presence of this septa also helps in the identification of the fungi, right? So mostly molds have septa, but not all the groups have septa, right? Okay. Now, if you look at the colonial structure of the fungi, those hypha which are extended out of the surface of the mold, they are called as aerial hypha, and those which form the baseline of the colony, they are the vegetative hypha. So You can also say this, that the portion of the hypha that obtain nutrients is called as the vegetative hypha. So they must be stick to the surface from where they need to absorb the nutrients, and those which are projected outward and they are mostly involved in the reproduction, so they are called as reproductive hypha, or they are called as the aerial hypha, because they are projected above the surface of the medium on which the fungus is growing. Right, so aerial hypha, they often bear reproductive spores, as you can see here. Okay, this is the aerial hypha on which these reproductive bodies are present. And vegetative hypha is that hypha which is actually from the baseline of the mycelium, or you can say the basal structure. Now moving towards one of the forms of the fungi, because we have discussed that how mold behaves when growing in the form of a colony. But yeast, usually when you look at the yeast, unicellular structures, they are actually either spherical in shape or they are the oval in shape, and their colonies are found as the white or creamy colonies, and on fruits and leaves their presence is shown by a white powdery coating. When you can identify easily by scraping the growth, and you can have a drop of lactophenol, cotton glue, and you can mix it with the spread it with the inoculum, and you can observe under microscope. So if you observe such a spherical structure and large, comparatively yeast size is larger than the bacterial cells, so they are usually representing the yeast. So yeast can be can be a budding yeast, or it can be a non-budding yeast, right? So budding yeast, one of the example is the Saccharomyces. It divides unevenly, as you can see that from a parent cell, first a small bud is formed, and that bud that it starts growing, and then later on sometimes it is separated from the parent cell, sometimes it is not separated from the parent cell. So sometimes budding is one of the method of reproduction in yeast. So one cell can in time produce up to 24 daughter cells by budding, right? And I told you that some yeasts, they produce buds which are filled to detach themselves, so they form short chains of cells, like short chains of spherical cells. So these are called as pseudohypha. They represent they mimic the hypha, but they are not the actual hypha. So that is because of the uh, non-detachment of the cells, right? Like I gave you the example here that Candida albicans is one of the yeast other than Saccharomyces, uh, which attaches to human epithelial cells as yeast, but usually requires pseudohypha to invade deeper tissues. Candida albicans is one of the pathogenic yeast that targets the many areas of your skin. So they require pseudohypha. And there are some other yeasts which are called as Schizosaccharomyces, which divide evenly to produce two new cells. There are different types of yeast, and their budding pattern is different, right? So sometimes this budding pattern and the presence of pseudohypha, it helps a lot in the identification of the yeast. So that's why it needs to be discussed here, right? Another group of fungi is the dimorphic fungi. Dimorphic means As its name indicates that it can display both type of morphologies. It can display means one species can display either yeast-like growth or the mold-like growth, as you can see in the picture as well. Right, so it is the characteristic of mostly pathogenic fungi, which can grow either as mold or as the yeast. Right, so mold-like forms, they produce vegetative and aerial hyphae, and yeast-like forms, they reproduce by budding. So it depends. It depends on what. When any fungi display mold-like growth or when it displays the yeast-like growth. So mostly this dimorphism, it's temperature dependent. Means when the things are ambient, means at ambient temperature, means the room temperature 37 degrees centigrade, fungus behaves like yeast. But whenever the temperature it goes down, so it behaves like mold, right? And one of the example is the mucor. As far as the nutritional adaptations are concerned, the first and foremost is their uh, acidophilic nature. Okay, so fungi, they grow much better in an environment with a pH of about 5, which is acidic for the growth of most common bacteria. So those foods which are acidic, they are mostly targeted by the fungi. For example, if I give you the example of fermented pickle, so mostly it is spoiled by fungal growth. Secondly, they are all aerobic. Molds are aerobic, and while the yeast forms, they may be aerobic or they may be facultative anaerobes. So this is also the difference. So mostly molds, they need oxygen. Um, if you want to save your pickle from the mold or moldy growth, just just dip it in the layer of oil. Okay, oil must be on the surface and all the content should be uh, beneath that surface if you want to protect your food from the acidic food from the mold growth. So another exceptional property is the resistance to osmotic pressure, because they are mostly resistant to high osmotic pressure and they can survive and the not survive rather they are osmophilic. They love to grow at high osmotic pressure. So comparative to bacteria, because bacteria, they are sensitive to high osmotic pressures. So that's why mostly fungi, they are able to grow at high salt or high sugar concentrations. Plus, fungi requires very low moisture content. So very low moisture content is sufficient enough to favor the growth of fungi comparative to the growth of bacteria. Bacteria require comparatively larger volumes or higher moisture content. Plus, nitrogen requirement is less than bacteria. And then fungi, I told you that they are capable of metabolizing complex carbohydrates such as lignin. Lignin is one of the toughest carbohydrate in this planet, which is a component of food. So fungi even can hydrolyze that lignin. So mostly bacteria, they cannot use it for the nutrients. So their nutritional adaptation is very vast. Their metabolization of the complex carbohydrate is very vast, diverse, I must say. So now moving towards its reproduction, because reproduction is that property that helps a lot in identification. So basically, the fungal strains, they are mostly identified by the types of reproductive structures they form. So both mode of reproduction, they can be observed both sexual and asexual reproduction in fungi can be observed, and it is because of the formation of different spores, sexual spores and asexual spores. So I told you that mostly fungi, they are identified by the spore type. And now if you are talking about the fungal spores, the fungal spores, they are very different from bacterial endospores. How they are different? Because the bacteria forms endospores as a method of their survival strategy, not as a method of their reproductive uh, cycle, right? So if a cell forms endospore, the parent cell has to sacrifice itself so that endospore remains there, right? So after the release of the endospore, the parent cell is no more, right? So endospore basically is a method of survival, and these endospores, they remain in the environment, and whenever uh, they found any suitable environment, they germinate themselves into uh, a, a new vegetative cell. But it doesn't increase any population size, right? Whereas fungal spores, they are formed in numerous quantities. They are formed as a means of reproduction. A parent cell generates huge quantities of spores while remaining itself, and while remaining by keeping itself alive, not like the bacteria. Secondly, the fungal spores, they are actually the true reproductive structure. They are not the method of survival, right? Fungal spores are produced as a reproductive cycle, during the reproductive cycle of the fungi, whether it is asexual spores or the sexual spores. The second thing is that mostly bacterial spores, if we compare the two, fungal spores and the bacterial spores, they are more resistant. They are more tolerant to the extreme conditions. Their lifespan is much longer comparative to the fungal spores. So they mostly fungal spores, they do not exhibit the extreme tolerance and longevity of the bacterial endospores. They are not as tough as the bacterial spores, right? Now, if we're talking about spores, fungal spores, so asexual spores definitely involves a single parent, so it is formed by the hypha of one organism. So when these spores germinate, they become organisms or that are genetically identical to the parent. And but whenever um, sexual reproduction is there, so two different hypha, they contribute towards the formation of a single spore. So those spores display properties of the both parents. Now, asexual spores are used for the identification of different fungal strains. So basically, you can broadly divide them into two types. It could be sporangio spores, or it could be conidiospores. So first we'll discuss the sporangiospores. The sporangiospores basically are formed, as you can see, um, they are formed into a sac, and which is called as a sporangium, and the hypha on which these sporangium they are located, these hypha are called as sporangiophores. So in this sporangium there are a lot of spores. So when a sporangium ruptures, a lot of spores they are liberated. So this is the common way of reproducing the new cells. So they are enclosed into basically enclosed into a sac which is known as a sporangium, and the hypha that bears that sporangium is known as sporangiospore, and those spores they are called as sporangiospores. And this is the example of rhizopas, which is given here. Okay. Now moving towards the conidiospore. Conidiospores, they are different from sporangiospores in that sense that they are not enclosed into any sac. They are produced at the ends of the hypha, at the tips of the hypha. So they are called as the conidiospores. The word conidia actually means dust. So they are producing so numerously, and they are produced so numerously, and they are so tiny, and they are so lightweight, lightweighted structures that they can be easily move from one place to other by means of air, just like the dust particles. So conidia behaves the same. That's why they are called as conidiospores. So conidiospores, they may be formed like this. At the end of the hypha, they form like these cells. They are separated by septa at the end of the hypha. So these spores are liberated. So they are called as conidia. So the hypha that generates conidia, that hypha is called as the conidiophores. An example, there are so many examples. Then, so this is the typical example of conidiospores, but conidia can be they can be not always they are displaying like this structure, but they may be of this type. They may be formed by these small fragments, fragmentation. So fragmentation of the hypha at the tips, it results in some this type of structures. They are also called as conidia, but they are different from this type of structure. So they are called as the arthroconidia, means those conidia which are formed by fragmentation, right? And there are some conidia which are formed at the end as just like the buds. So they are called as the blastoconidia, which are formed from the buds of the parent cell. So conidiospores can be of many types. Okay, they are called as blastospores. They may be called as arthrospores or arthroconidia or blastoconidia or blastospores. And there are some other conidiospores like which are formed at the within the hypha, and they are the actually tough cellular structure with thick cell wall, which are different from these all three types, because these three types they are formed at the tips, but this is the structure which is not found at the tips but is formed in between the hyphal structures. So chlamydoconidium or chlamydospores, it is also a thick walled cells within hypha, right? They may be separated, and chlamydospores, they are liberated. So presence of chlamydospores, they are the characteristics of some hypha, characteristic of some species. So there are different conidiospores which are formed by different species of fungi.